To understand why some people develop aggressive cancers, we may need to look beyond genetics alone. Most people believe cancer is purely genetic, that DNA damage is irreversible once it occurs. Many assume that once cells turn cancerous, there's nothing that food or nutrition can do about it. But research and clinical experience suggests something that challenges what many learned in medical school. Certain foods may actually help repair damaged DNA and limit tumor growth by affecting blood supply. A microscopic tumor without access to blood vessels cannot grow larger than the tip of a ballpoint pen. But once blood vessels reach that tumor, research suggests it can expand dramatically in just 2 weeks. To understand why this matters, we need to explore the concept of angiogenesis and what it means for cancer. Angiogenesis simply refers to the growth of new blood vessels throughout the body's tissues and organs. Your body carefully regulates this process, since you need just the right number of blood vessels at all times. Too few blood vessels and tissues begin to suffer from lack of oxygen and essential nutrients over time. Too many blood vessels can lead to problems, such as the abnormal growth seen in conditions like diabetic eye disease. The body maintains a careful balance, promoting blood vessel growth when needed and stopping it when sufficient. Cancer cells can disrupt this balance by hijacking the body's angiogenesis system for their own benefit. Tumor cells release proteins called growth factors that stimulate new blood vessels to grow directly into them. Once those blood vessels connect to a tumor, the rate of growth can change in a significant and measurable way. Research has shown that a tumor completely isolated from blood supply stays very small, around 1 to 2 millimeters. That size is smaller than a grain of rice, and such a tumor can remain essentially harmless for years or even decades. But once angiogenesis occurs and blood vessels reach that tumor, growth may accelerate in an extraordinary way. Research from Harvard Medical School tracked tumor growth and found that vascularization dramatically increases size. A tumor the size of a pinpoint could potentially become the size of a grape within approximately 2 weeks of receiving blood. This is one reason why early detection matters and why preventing tumor angiogenesis is a focus of cancer research. Pharmaceutical companies have invested significantly in developing drugs designed to block tumor angiogenesis. These drugs, called angiogenesis inhibitors, are now used as standard treatments for a number of different cancers. They work by cutting off the blood supply to tumors, essentially limiting the nutrients those tumors can access. However, these drugs can have significant side effects because they affect blood vessel growth throughout the entire body. In laboratory research, scientists began testing not just pharmaceutical compounds, but also extract from common foods. When green tea extract was tested using the same methods used to evaluate experimental drugs, the results were notable. The polyphenol in green tea, particularly a compound called EGCG, showed measurable anti-angiogenic activity in the lab. Researchers noted the results were striking enough that, had it come from a pharmaceutical source, trials might have followed. Green tea is not the only food showing this type of activity in laboratory settings when carefully tested and measured. When foods are tested using rigorous scientific methods, over 100 have shown measurable anti-angiogenic properties. These foods may slow or inhibit the growth of blood vessels that could otherwise feed developing tumors in the body. This is not speculation, but is measured in laboratory assays where blood vessel growth can be directly observed. A second important mechanism involves the ability of certain foods to support the repair of damaged DNA and cells. Every single day, your DNA is exposed to damage from ultraviolet radiation, environmental pollutants, and metabolic processes. Your cells have repair mechanisms that function like a maintenance crew, fixing problems before they become permanent mutations. However, those repair systems need specific nutrients to function at their best over time. Without adequate nutritional support, DNA damage may accumulate in ways that could eventually contribute to disease. Think of it like a road developing potholes. Without the right materials and a working crew, damage builds over time. Some of the most compelling research on food-based DNA repair comes from studies conducted using kiwi fruit. A study from the University of Oslo recruited 43 healthy adults and had them eat 1, 2, or 3 kiwis daily. After 3 weeks, researchers measured DNA damage in participant cells using a technique called the comet assay. This test makes broken DNA strands visible under a microscope, where they appear like comets with trailing tails. Participants eating 1 kiwi daily showed 60% less DNA damage compared to their measurements at the start. Those eating 3 kiwis daily showed not only prevention of new damage, but also repair of existing DNA damage. Their cells showed a 60% improvement in DNA repair capacity by the end of the 3-week study period. The mechanism may be connected to vitamin C and other antioxidants found in concentrated amounts in kiwi fruit. Vitamin C can neutralize free radicals that cause DNA strand breaks and may also support enzymatic DNA repair systems. Kiwis also contain polyphenol, flavonoids, and carotenoids that may work together in ways researchers are still exploring. This brings up an important point about whole foods compared to isolated supplements taken in pill or capsule form. A vitamin C supplement provides antioxidant benefits, but eating a kiwi provides vitamin C alongside many other compounds. Those compounds may enhance each other's effects in ways that isolated supplements are not able to replicate alone. Most prevention advice focuses on avoiding carcinogens, such as not smoking, limiting alcohol, and reducing sun exposure. Those measures do matter, but there is also value in actively supporting the body's existing anti-cancer defense systems. Your body is already designed with immune cells, blood vessel regulators, and DNA repair mechanisms to address abnormal cells. The question is whether those systems are receiving the nutritional support they need to function as effectively as possible. The first anti-cancer pathway that food may support involves anti-angiogenesis, as discussed earlier in this explanation. Foods containing certain polyphenol and flavonoids may help inhibit the growth factors that tumors release to attract blood vessels. When tumor cells release VEGF, a protein that signals for new blood vessel growth, certain food compounds may block that signal. The second pathway involves direct support of the immune system, including cells called natural killer cells and T cells. These immune cells are among the body's primary tools for identifying and eliminating abnormal and potentially cancerous cells. A study from the University of Auckland examined immune cell counts in athletes who consumed blueberries over 6 weeks. Athletes are useful subjects because intense exercise temporarily suppresses immune function, making dietary effects easier to detect. After consuming 1.5 cups of blueberries daily for 6 weeks, athletes showed 88% more T cells. Their natural killer cell activity was also significantly elevated compared to measurements taken at the beginning of the study. The active compounds responsible for this response are anthocyanins, the blue pigments that give blueberries their distinctive color. A third pathway involves the connection between gut bacteria and immune function, an area of growing scientific interest. Your gut contains trillions of bacteria, and certain species appear to directly influence how well the immune system performs. One bacterium in particular, called Akkermansia mucinifila, has emerged as a notable player in immune and cancer research. Research from the Gustave Roussy Cancer Center in France studied 200 cancer patients receiving immunotherapy treatment. Immunotherapy is a treatment designed to help the immune system recognize and attack cancer cells more effectively. It works very well for some patients, but for others, it provides little or no measurable benefit during treatment. Researchers analyzed stool samples from all 200 patients to understand what might explain the difference in outcomes. The most significant difference between responders and non-responders was the presence of Akkermansia bacteria in the gut. Patients with high levels of this bacterium responded much more strongly to immunotherapy than those with low or absent levels. This bacterium appears to prime the immune system in ways that may make cancer treatments more effective for some patients. Understanding these mechanisms helps explain why certain foods may offer more than general nutritional benefit over time. The first food to consider is kiwi fruit, with a general recommendation of 1 to 3 kiwis consumed daily. For general DNA protection and maintenance, 1 kiwi each day may be sufficient based on the available research findings. Those with higher risk factors or a personal or family history of cancer may benefit from aiming for 3 kiwis daily. Research suggests that maximal benefit in the DNA repair studies was observed at the 3 kiwi per day consumption level. Kiwis can be eaten with their skin on if organic and thoroughly washed, as the skin provides additional fiber and polyphenol. Golden kiwis have smooth skin and are considered just as nutritionally beneficial as the more common green variety. Kiwis can be sliced, added to yogurt, or blended into smoothies as practical ways to include them in a daily routine. They are also a source of prebiotic fiber that may support the growth of beneficial bacteria in the gut microbiome. The second food is green tea, with a general recommendation of 3 cups of properly brewed tea each day. Bottled green tea products from convenience stores often have minimal active compounds and may contain added sugar. Brewed green tea made from actual leaves is the preferred form for obtaining meaningful amounts of active compounds like EGCG. EGCG has been associated with inhibition of tumor angiogenesis and has been studied for other potential anti-cancer effects. A study from the Chinese University of Hong Kong followed 68,000 women over more than 10 years. Women who drank green tea regularly, defined as at least 3 cups per week, had a 27% lower colorectal cancer risk. Those who drank green tea daily showed even more favorable outcomes compared to those who rarely or never consumed it. To brew green tea effectively, heat water to approximately 175 degrees Fahrenheit, or just below boiling. Steep the tea for 3 to 5 minutes, noting that longer steeping extracts more EGCG, but also produces a more bitter flavor. Adding lemon juice may increase EGCG absorption by roughly 6 times, because vitamin C helps stabilize the compounds during digestion. Dairy milk proteins may bind to the beneficial compounds in green tea and reduce their absorption, so it is best avoided. Plant-based milks, such as almond or oat milk, can be used as an alternative if you prefer a lighter tea. The third food is blueberries, with a general recommendation of 1 cup consumed daily, either fresh or frozen. Frozen blueberries are sometimes higher in anthocyanins because they are picked and frozen at peak ripeness for preservation. Anthocyanins may boost immune cell production, reduce certain types of inflammation, and have anti-angiogenic properties as well. Blueberries appear to reduce harmful chronic inflammation while simultaneously supporting beneficial immune responses against abnormal cells. A study from the University of East Anglia tracked nearly 200,000 people over more than 20 years. Those who consumed the highest amounts of anthocyanin-rich foods, primarily berries, showed a 17% lower overall cancer risk. For specific cancers such as esophageal and colorectal cancer, the observed risk reduction in that study was even higher. The fourth food surprises many people because there has been considerable misinformation surrounding it over recent years. Soy foods, including tofu, tempeh, edamame, and soy milk, are associated with meaningful anti-cancer properties in the research. A common concern involves phytoestrogens in soy, which some claim mimic human estrogen, but this understanding is not accurate. Phytoestrogens are structurally different from human estrogen at the molecular level and may actually block estrogen receptors. This receptor blocking effect may be protective against hormone-related cancers, such as breast cancer in certain contexts. The active compound in soy called genistein is one of the more extensively studied anti-angiogenic substances in food science. Research from the University of Illinois found that genistein inhibited VEGF signaling by over 70% in experimental models. Population data also shows that regions with high soy consumption tend to have lower rates of breast and prostate cancer. A meta-analysis combining data from 35 studies found that women with the highest soy intake had a 29% lower breast cancer risk. Women already diagnosed with breast cancer who consumed more soy also showed associations with better outcomes in some analyses. The fifth food is broccoli, and specifically broccoli sprouts, which are significantly more concentrated in the active compound. The key compound is sulforaphane, which activates the body's detoxification systems and has been studied for direct anti-cancer effects. Mature broccoli contains sulforaphane, but broccoli sprouts, the 3 to 4 day old seedlings, may contain 100 times more. Research from Johns Hopkins University found that a small handful of broccoli sprouts may provide as much sulforaphane as several pounds of mature broccoli. Broccoli sprouts can be found in the produce section of many grocery stores or grown at home in a jar within about 4 days. Eating 1/4 cup of broccoli sprouts 3 to 4 times per week is a practical starting point based on available research. If sprouts are not available, regular broccoli remains beneficial, with the stems containing roughly twice the sulforaphane of the tops. Lightly steaming broccoli for 3 to 4 minutes may increase sulforaphane bioavailability compared to eating it completely raw. Boiling broccoli, however, can destroy sulforaphane, so steaming or light cooking is generally the preferred preparation method. The sixth food is tomatoes, and specifically cooked tomatoes, which provide greater amounts of the active compound lycopene. Lycopene is a carotenoid that gives tomatoes their red color and has been studied for anti-angiogenic and DNA protective effects. Cooking tomatoes breaks down cell walls and releases lycopene in a form that the body can absorb much more efficiently. Tomato sauce, tomato paste, and roasted tomatoes all provide significantly more bioavailable lycopene than raw tomatoes do. A landmark study from Harvard followed over 47,000 men for 12 years and examined tomato consumption patterns. Men who consumed tomato products at least twice per week had a 23% lower risk of developing prostate cancer. Those who consumed 10 or more servings of tomato products weekly showed even greater risk reduction, up to 35%. Aim for at least 3 servings per week of cooked tomato products, such as half a cup of sauce or 1 cup of cooked tomatoes. Adding olive oil when cooking tomatoes may enhance lycopene absorption, since lycopene is a fat-soluble compound. The seventh food is pomegranate, with a general recommendation of about 4 ounces of pure juice or half a fresh pomegranate daily. Pomegranates contain elagitannins that gut bacteria convert into urolithin A, a compound associated with cellular and immune health. Pomegranate may also feed Akkermansia mucinifila, the bacterium that has been linked to better immunotherapy outcomes in research. One clinical example describes a patient whose Akkermansia levels were restored through regular pomegranate juice consumption over weeks. After rebuilding her gut bacterial levels, she reportedly responded very strongly to subsequent immunotherapy treatment for her cancer. While this represents one individual case, it illustrates the broader research interest in using food to support medical treatments. For daily prevention, 4 ounces of pure pomegranate juice without added sugar or half a fresh pomegranate may be sufficient. Elagitannins can also be found in walnuts, raspberries, and cranberries, allowing for variety while maintaining potential benefits. Timing may play a role in how effectively these foods support the body's natural defense and repair systems over time. DNA protective foods like kiwi and berries eaten earlier in the day allow more time for nutrient absorption before sleep. Anti-angiogenic foods like green tea and soy may be better suited to afternoon or evening consumption based on these considerations. Avoiding heavy meals within 3 hours of bedtime may also help because active digestion can divert resources from cellular repair. Combining certain foods may create synergistic effects that are more powerful than consuming those foods individually and alone. Green tea with lemon may increase EGCG absorption 6-fold, while tomatoes cooked with olive oil may triple lycopene absorption. A smoothie with blueberries, kiwi, and spinach provides anthocyanins, vitamin C, and sulforaphane compounds all at the same time. A meal of baked tofu, steamed broccoli, and tomato sauce combine genistein, sulforaphane, and lycopene in a single serving. Consistency is more important than occasional large amounts when it comes to activating and maintaining protective dietary pathways. 1 cup of blueberries every day is likely more effective than 7 cups consumed all at once in a single sitting. Regular daily or near daily consumption creates sustained activation of the protective systems that rely on consistent nutritional input. Think of it like maintaining a fire. Steady, consistent fuel keeps it burning more reliably than sporadic large additions do. One common mistake is believing that perfect eating is required and then stopping entirely when that standard cannot be maintained. The body has redundant defense systems, and consistently eating most of these foods most days provides meaningful ongoing support. This approach is about empowerment and consistent positive habits, not about fear, deprivation, or achieving absolute perfection. Another mistake is assuming that supplements can fully replace the benefits provided by whole foods in their natural form. Lycopene capsules, EGCG extracts, and anthocyanin supplements may have a role, especially when certain foods are not accessible. However, supplements provide isolated compounds, while whole foods provide hundreds of compounds that may work together synergistically. A tomato contains lycopene alongside vitamin C, potassium, folate, and other carotenoids that may enhance lycopene's overall effects. Preparation methods can significantly affect how much of the active compounds your body actually absorbs and is able to use. Cooking tomatoes, lightly steaming broccoli, and using the right water temperature for green tea all make a measurable difference. Paying attention to these practical preparation details may improve outcomes without requiring major changes to eating habits overall. Focusing on adding beneficial foods rather than strictly eliminating beloved ones tends to support longer-term dietary consistency. Adding blueberries to breakfast and having green tea in the afternoon is a sustainable habit for most people over time. As more protective foods are added, they naturally begin to crowd out less nutritionally supportive options in the daily diet. This gradual and additive approach is far more sustainable over years and decades than restrictive elimination-based diets tend to be. Cancer does not have to be viewed as an inevitable part of aging based on what the research increasingly suggests. People who remain cancer-free or recover from cancer are not necessarily genetically different or uniquely fortunate in their outcomes. Many consistently eat foods that may help activate the body's natural defense systems against abnormal cell growth over time. Your immune system can recognize and eliminate cancer cells. Your body can regulate blood vessel growth to limit tumor expansion. Your cells also have the capacity to repair damaged DNA when they receive the nutritional support needed to function well. To summarize, cancer cells need blood vessels to grow beyond microscopic size, and food may help limit that process. Foods containing specific polyphenol and flavonoids may help block angiogenesis and limit the blood supply that tumors require. Certain foods, particularly kiwis, may provide meaningful DNA protection and support the repair of existing DNA damage in cells. Gut bacteria, especially Akkermansia mucinifila, appear to play a role in how effectively the immune system responds to cancer. The 7 foods discussed include kiwis, green tea, blueberries, soy foods, broccoli sprouts, cooked tomatoes, and pomegranate. These foods may work through multiple mechanisms, including anti-angiogenesis, immune activation, DNA repair, and microbiome support. Consistency, proper preparation, and combining foods thoughtfully may enhance the protective effects beyond what any single food provides. You may have more influence over your long-term cancer risk than is commonly understood by consistently supporting your body's defenses.